oxidative stress in goats. Here, oxidative stress not happens only in goats. It happens all living things that are found on the earth. In this topics, we covered the what is the oxidative stress and how it affects the productive and reproductive efficiency of the goats, and how to manage the oxidative stress. All these things are covered, have been covered in this topics. First of all, goat can be grown with minimum effort without a large investment. Provide maximum output through milk and meat and wool, and support marginal farmers. Small ruminants are rare, that is the beneficial, because the small ruminants having short generation intervals, multiple birth, and accelerated growth that easily adapted to the low quality of the forage. Goat is rare for the milk, meat, manure, fibers. All are these productive performance are there are several factors that are affected these productive performance and reproductive performance of goat. One of these, the oxidative stress. What is the oxidative stress? The knowledge of oxidative stress is important because it damages the not only cell, protein, DNA, and have negative impact on the growth, immunity, and reproduction. It also makes prone to the goat for the occurrence of the metabolic and infective diseases. Oxidative increase the mastitis and somatic cell count in the milk, and also decrease the fertility, increase the embryo mortality and postpartum retained of placenta, and decrease this oxidative stress. Not only decrease the shelf life of meat, but also decrease the shelf life of the milk. Then what is the oxidative stress? In these pictures, it has been shown that this one is the situation of the oxidative stress. When the oxidative stress occurs, when the imbalance between the free radicals and antioxidant that are present in the body, either increase the production of reactive oxygen species or depleted the antioxidant level in the body. Both the condition is cause oxidative stress. This one is the normal situation. This situation is the balanced by these free radicals are balanced by the antioxidant, and some free radicals are some concentration, low or moderate concentration of the free radicals are the beneficial for the body. But when the concentration increase excessively greater, then cause the oxidative stress. Reactive oxygen species or nitrogen species. This one is the free radicals. This one is the free radical, that is the derived either oxygen molecules or nitrogen molecules, and the free radicals are most reactive in the nature, and having one or more unpaired electron in the last orbit, and they want to relieve their unpaired electron by reacting the micromolecules. This is the examples of the reactive oxygen species and reactive nitrogen species, and these are the produced in the body. That is the harmful. Not always harmful, only low and moderate concentration is beneficial, but excessively produce the production of reactive oxygen species has a harmful effect. Actually, this one is the free radicals. The free radicals having one unpaired electron and always want to act as a thief and want to steal electron from another healthy molecule. But if the sufficient concentration of the antioxidant are present in the body, then these are the donates the electron and makes the free radical in normal healthy cells. There are two sources of the free radicals formation. First one is the endogenous sources. Second one is the exogenous sources. These are the endogenous sources. Peroxisome, mitochondria, immune cells, that is neutrophils, macrophages, plasma membrane, cytoplasm, endoplasmic reticulum, all are the responsible for the production of reactive oxygen species. Mitochondria acts as a major source for the production of reactive oxygen species and alone produce about 90%. And these enzyme systems in the mitochondria is the responsible for the production of the reactive oxygen species. The superoxide radicals generated in the mitochondria is neutralized into the hydrogen peroxide by the presence of superoxide dismutase that are present in the mitochondria. This peroxide, the hydrogen peroxide, after Penton reaction, this change into the hydroxyl radical, and this hydroxyl radical are very, very harmful as compared to all other free radicals. This is the mitochondria by which in this picture it has been shown that how the free radical generation takes place in the mitochondria. This one is the picture shows that the complex first that I told in the previous slide, the complex first and complex seconds electron leaks and oxygen molecule change into the superoxide radicals. This is the enzyme systems. These are the actually this one is the complex first and complex third, where the electron leak out and form the free radicals, superoxide radicals. Free radicals. And this one is the non-enzymatic. This one is the found the non-enzymatic reaction and cytochrome 450 xanthine oxidase also found in the cytoplasm that are also responsible for the production of the free radicals. And this NADPH oxidase present on the cell membranes. This one is also responsible for the production of the reactive oxygen species. Peroxisome. Second one is the peroxisome. That one is the also a major site for the production of the hydrogen peroxide and beta oxidation. The enzymes that takes place in the beta oxidation are capable to produce the hydrogen peroxide in the peroxisomes. And this a part of this, the peroxisome also producing radicals like the superoxide radicals, hydroxyl radical and nitroxyl radicals, and NADPH oxidase that are found on cell membrane are responsible for the production as discussed in the mitochondria, and xanthine oxidase that are found in the cytoplasm will also produce the free radicals. This is the pictures in of peroxisome that in this picture it has been shown that how the radicals are produced in the peroxisome. Third one is the endoplasmic reticulum. The endoplasmic reticulum is also responsible for the production of the hydrogen peroxide and other free radicals also. This is the plasma membrane. This one is the this structure is the NADPH oxidase that is responsible for the production of the free radical superoxide free radicals from the oxygen in the plasma membrane. Second one is the exogenous source. What is the exogenous source and which one is the responsible? And this one is the drought condition. Drought condition, nutritional deficiency, exposure to herbicide and pesticide, exposure to heavy metals, peri-parturient period, heat stress, cold stress, and salinity. All these factors are responsible for the production of the free radicals that increase the free radical production. These factors increase the production of free radicals, and these cause the oxidative stress in the goats. The drought condition, the forage grow in the drought condition, that is the poor in antioxidant. If the fodder is the poor in the antioxidant, then the antioxidant level is the decrease in the goat blood due to the dietary content of the antioxidant level are decreased. And the goats that have fed on green forage, they are their blood contains high antioxidant level, because the dry forage is very rich in the green forage is rich in the antioxidants. Peri-parturient period. What is the peri-parturient period? The period between the 3 weeks before and after calving is called peri-parturient period. This one is the very crucial period for maintaining the health and productive performance. And dry matter intake during this period is decreased. Therefore, nutrient demand necessary for the fetal growth and lactogenesis is decreased, resulting in negative energy balance. And this metabolic adaptation under the negative energy balance cause adipose tissue breakdowns in the form of the NAFA, non-esterified fatty acids, that triggers the generation of the free radicals formation during the peri-parturient period. Then next one is the heat stress. High ambient temperature and relative humidity can be cause oxidative stress in the goat. This is the THI value by which THI here, the temperature humidity index, is calculated by the this formula. And when the value is the less than or equal to 72, then the animal feel comfort. And if the value is the 73 to 78, then the goats feel the moderate heat stress. If the THI value greater than the 78, then the animal feel severe heat stress. And heat stress also promotes the production of the free radicals in the different cell organelles. This is the effect of the exogenous sources and antioxidant status of the goats. The Muhammad et al. 2020 reported high salt intake in goat increased the free radical formation by increasing the activity of the NADPH oxidase enzyme that are found on the plasma membrane. And this antioxidant levels, that is the superoxidase mutase catalase and glutathione peroxidase, decrease during high salt intake in the case of the goat. And Gupta et al. 2021 reported heat stress is also produced the greater concentration of the free radicals and decrease the antioxidant level in their blood, that in the blood of that goats, that is heat stress. The Sally et al. 2008 also reported that peripartum period in the goats promotes the production of the free radicals and decrease the antioxidant level during these period. Either animal are in heat stress or in periparturient period or continuous in stress condition, and these conditions are promote the production of the free radicals, and goats during these conditions are continuous in oxidative stress. The role of the free radicals. The role of, if physiological role. If the concentration of the free radicals low or moderate, then the these free radicals have beneficial effects and improve the immunity. Improve the immunity by which phagocytic cells like the neutrophil and macrophages synthesize the free radicals in their cytoplasm, and when any microorganism, foreign microorganism enters into the body, then these free radicals are utilized to kill the microbes. And this not only improve the immunity, this also cause the cellular signaling. Cellular signaling, that means cell communicated by one another by the free radicals. Nitric oxide, that is the cell to cell messengers, and proper blood flow modulation and normal neural activity, uh, and also involves in the whole depends by the eliminating the intracellular pathogens, and not only cellular intracellular pathogens that comes from outside, that these the free radicals are also help in the elimination of the tumor cells. And this free radicals, when the normal concentration, that also involves in the mitosis of the cell. A part of these, the free radicals in normal concentration, then it also helpful in the capacitation, hyperactivation, ectosome reaction, and fertilization. Then here capacitation and hyperactivation, ectosome reaction, and fertilization process occur in the female reproductive tract, by which the spermatozoa gains the ability to penetrate the oocyte. Without this capacitation, hyperactivation, ectosome reaction, the spermatozoa are not capable to penetrate the oocytes. Therefore, the free radicals are very important in the capacitation and hyperactivation, ectosome reaction, and fertilization when their concentration is not. This is the mechanism by which the free radicals are promotes the activation and hyperactivation and other fertilization process. Second one is the pathological effect of the reactive oxygen species. The exogenous and endogenous source promotes the production of free radicals in the goats that cause the oxidative stress. If their concentration is excessively increased, then cause oxidative stress, and these free radicals are able to damage the protein, lipid that are present in the cell membrane, because the polyunsaturated fatty acid are very rich in the cell membrane. Therefore, this lipid are very vulnerable to free radicals. Not only degenerate protein and lipids, but also in the mitochondrial DNA and cell DNA. These all the factors cause the cell death and cause the eventually cause the disease development and disease development in the either metabolic or infective diseases happens in the goats. This is the oxidative stress. In this picture, it has been shown that how the oxidative stress damage the lipid, protein, and DNA, how the normal cell change into abnormal cells and cause some are apoptized, and some cells are become necrotic, and this may cause cancer also. The plasma membrane of immune cells and germ cells. Immune cells like leukocytes, neutrophils, macrophages, T and B cells are very rich in the polyunsaturated fatty acids, and germ cells are also very rich, either oocyte or spermatozoa are very rich in the polyunsaturated fatty acid, and this is the site. In this picture, it has been shown that how the spermatozoa are degenerated by the free radicals. free radicals. This is the DNA. DNA is damaged by the, as I told in the earlier slide, the DNA damaged by the free radicals, and mitochondria itself and mitochondrial DNA is also damaged by these free radicals. This is the site of the, and the plasma membrane that is the found around the tail. This one is the also the damage, and this cause the infertility in case of the males. This is the diagrammatic representation how the cause damage the male spermatozoa and cause male infertility in the male. Oxidative stress cause the increase the production of reactive oxygen species, and this cause the lipid peroxidation, and this lipid peroxidation impair the motility and impair the fertilization, and this reactive oxygen species cause the DNA damage, and all these factors are responsible to cause the male infertility in case of the male goats. Lipid peroxidation, how to free radicals damage the lipids. Lipids are present in the plasma membrane in the form of the polyunsaturated fatty acid. Polyunsaturated fatty acid are extremely vulnerable to the free radicals, and these lipids may disturb normal function of the cells, and the biomarker of the lipid peroxides, lipid peroxidation is the lipid peroxide is the best biomarker of the oxidative stress. This is the how to initiate the chain reaction. The lipid reacts with the free radicals. Free radicals react with the lipids and to form the lipid radicals, and these lipid radicals in the presence of the oxygen to form the lipid peroxy radicals. This is the chain reaction. This and this lipid peroxy radicals again bind with the lipids to form the lipid free radicals. Lipid free radicals, and these free radicals again and again bind to the lipid molecules and damage the whole membrane. But if the concentration of the antioxidant are present in the body, that are the neutralize the lipid peroxide radicals, and itself form the alpha tocopherol radical, and binds this alpha tocopherol radical binds with the peroxy radical, lipid peroxy radical to form the non-radical product. Means here this antioxidant, how to antioxidant is the beneficial to minimize or neutralize the free radicals concentration in the body. There is how to diagnose the oxidative stress. There is no clinical sign of the oxidative stress that is observed in the animals, but the condition like periparturient period, oxidative stress, and the young ones animals are the very feel the oxidative stress during this period, and we observe these by lab diagnosis by the this one is the malondialdehyde, thibarbituric acid reactive substance, myeloperoxidase. These are the end product of the lipid peroxidation. Malondialdehyde, this is the biomarker of lipid peroxide, and myeloperoxidase is found in the neutrophils that promotes the production of the free radicals. This one is the reactive oxygen species that I told in earlier slide. Their concentration is increased excessively, then cause the oxidative stress, and this one is the biomarker of the oxidative stress, and this one is the superoxide dismutase and catalase and glutathione peroxidase. These are the antioxidant enzymes that are the also their level is decreased during if animal are in condition of the oxidative stress. This is the oxidative stress that I told in the earlier slide, that lipid and protein and DNA damage. These are the malondialdehyde and barbituric acid reactive substance. These are the biomarker of the lipid lipids and proteins. The biomarker carbonyl levels increase. This one is the biomarker of the proteins, and 8-hydroxy 2-deoxyguanosine is the also the biomarker of the DNA damage. And if their level is increased in the body, then means the animal condition of the oxidative stress. Here, antioxidant. What is the antioxidant? How it neutralize or decompose the oxidative free radicals and makes the animal normal and healthy. And this one is the either supplemented. Some are the endogenous antioxidants, some are the exogenous antioxidant. When we supplemented exogenous antioxidant, then these exogenous antioxidant improve the endogenous antioxidant level. First one is the what is the antioxidant? How it decompose the free radicals by 3 modes. And first one is the degenerating or neutralizing the free radicals or reducing their formation, or opposing their action. By 3 modes, the antioxidant protects the animal from the free radicals. The antioxidants are categorized into the 3 category: enzymatic and endogenous enzymatic antioxidant, endogenous non-enzymatic antioxidant, and exogenous antioxidants. Endogenous enzymatic antioxidant protects the DNA and mitochondrial DNA from oxidative damage, and also neutralize the free radicals before reacting with the biomolecules. And first one is the endogenous enzymatic antioxidant is the superoxide dismutase, and this superoxide dismutase are 3 types that is found in the cells. That is the copper zinc superoxide dismutase that is found in the cytosol of the cells, and manganese superoxide dismutase found in the mitochondria of the cells. And whereas the extracellular superoxide dismutase found in the extracellular fluid that protects the animal from the free radicals. This one is the very superoxide dismutase is the very important to neutralize the superoxide free radicals to the hydrogen peroxide and oxygen molecules. This one is the superoxide dismutase always compete with the nitric acid for the superoxide anion, prevent the formation of the peroxynitrate formation, because this nitric acid are the responsible for the formation of the free radicals. That is the derived reactive nitrogen species. And this superoxide dismutase change the superoxide anion to the hydrogen peroxide by this copper zinc superoxide dismutase. The second endogenous enzymatic antioxidant is the catalase, that is the heme oxidoreductase, and highest activity observed in the liver, and mainly synthesized in peroxisome. And 1 mole catalase responsible for the neutralize of the 6 into 10 to the power 6 mole of H2O2 per minute. And magnesium iron acts as a cofactor for their activity of the catalase. Without these minerals, the activity is impaired and not able to act. And this catalase change the hydrogen peroxide that are formed from superoxide radicals in the presence of the superoxide dismutate, and this hydrogen peroxide change into the H2O, the water, not a H2O. This one is the change hydrogen peroxide change into the hydrogen water and oxygen molecules. The catalase is responsible for the conversion. And third one is the third endogenous antioxidant enzyme is the found in the mitochondria and the cytosol. And selenium is acts as a cofactor for the activity of this enzymes. And this enzyme is also responsible for the conversion of the hydrogen peroxide to water and water. These glutathione molecules two glutathione molecule reduced form of glutathione molecule change into the oxidized glutathione molecules. Next is the ceruloplasmin. Ceruloplasmin is the glycoprotein that is found in the liver. The copper carrying, this one is the copper carrying protein, and the copper carrying protein that is the binds with the about 95% copper found in this ceruloplasmin in the form of ceruloplasmin, and this ceruloplasmin is the responsible for the iron metabolism, and this one is acts as a ferrooxidase and potent inhibitor of the myeloperoxidase that is found that I told myeloperoxidase found in the neutrophils that promotes the production of the reactive free radicals in the neutrophils, and this is the potent inhibitor. The ceruloplasmin is the potent inhibitor of the myeloperoxidase. That this enzyme is the responsible for the production of the lipid hydroperoxides during the inflammation. And this also ceruloplasmin is also change the ferrous form of to ferric form iron and prevent the Fenton reaction. And this hydrogen peroxide never change into the hydroxyl radicals because this one is the potent inhibitors of the Fenton reaction. This is the mechanism of action by which enzymatic antioxidant takes place. How this is the summary of the enzymatic antioxidant which these enzymes are acts. This is the hydrogen oxygen molecule, how to change the superoxide radical, and these enzymes that are responsible for the conversion of the oxygen into the superoxide radical, and these superoxide radicals change into the hydrogen peroxide in the presence of superoxide dismutase, and this hydrogen peroxide change into the water in the presence of catalase, and glutathione peroxidase is also responsible for the conversion of the hydrogen peroxide water, and makes these enzymatic antioxidants converts the free radicals into the non-free radicals. Non-enzymatic, some are non-enzymatic antioxidant. Glutathione is the this is the glutathione. This one is the also acts as a free radical scavenger, and this one is the distributed cytosol and nuclei, peroxisome and mitochondria. This liver is the main site for the synthesis of glutathione. The next one is the alpha lipoic acid. Alpha lipoic acid is also acts as a free radical decomposer, and this important thing is that this alpha lipoic acid is also regenerate the free radicals that form during the neutralization process. That is the alpha tocopherol radicals. This one is the regenerated by the alpha lipoic acids. And metal this one is the promotes the metal chelation and prevention the Fenton like radical reaction by which this one is the also prevent the conversion of the hydrogen peroxide to hydroxyl radical. And this alpha lipoic acid is synthesized in the mitochondria. Coenzyme Q10, this is present in the cell membrane, essential in the electron transport systems. And liposoluble, and this one is the acts as a liposoluble antioxidant, protect the lipid and protein protection from the free radicals. And quinol is the active form of the coenzyme Q10, and this coenzyme Q10 decomposes the reactive oxygen species and regenerate the oxidized antioxidant vitamin C and vitamin E. These are the exogenous antioxidants. These antioxidants are actually we are supplemented through diet, and these supplementation improve the endogenous antioxidants because some minerals are the acts as a cofactors for the enzymatic antioxidants, and some vitamins are also promote the production of the endogenous antioxidant. Therefore, this portion is the very important for the input part. Vitamin E. Vitamin E is the very important antioxidant that can be that may be supplemented through the diet, and this one is the rich in the green fodder, and but this vitamin E supplemented through diet is during the periparturient period heat stress and in growing kids may be supplemented. And this one is the chain breaking antioxidant and major membrane bound antioxidant in the cells, and one is the triggers the increase the apoptosis of the cancer cell, and in case of the human inhibit the free radical formation in the body. Vitamin C is also called ascorbic acid. It's water soluble, and this one is the acts as an antioxidant vitamin, but this one is the not given by the supplementation because these vitamins are synthesized in the in case of the goats, and because synthesized in the goats, and primarily is quenching the oxygen free radicals. Oxygen is quenching the free radicals that are derived from the oxygens and convert the vitamin free. This one is the also responsible for the conversion of alpha tocopherol radicals into the alpha tocopherol. Carotenoids. Without the carotenoids are the either this one is the two types that carotene or xanthophyll. If the oxygen without oxygen, that called the carotenoid, which called carotene. If contains oxygen, then called the xanthophylls. And this xanthophyll, this carotene, the example of carotene is the beta carotene and lycopenes, and xanthophyll. The example of xanthophyll is the astaxanthin. Beta carotene is the most diffuse carotene, and this one is the acts as a precursor for the vitamin A, and this one is the hunts the peroxyl, hydroxyl, and superoxide radicals. This one is the very important, and this may be supplemented through diet because this one is not synthesized in the goat's body. Xanthophyll, astaxanthin. Astaxanthin is the very important because this one is the most potent antioxidant that are known in the nature. This one is the most potent, and this one is the xanthophyll carotenoid, and obtained from the Haematococcus and Pythia rhodozyma. This Pythia rhodozyma is the yeast, and this one is the Haematococcus is the micro green algae, and this one is the most powerful antioxidant, and this one is the more stronger than the beta carotene, about 10 times stronger than beta carotene and 100 times stronger than the vitamin E. And therefore, this astaxanthin is called super vitamin E, and this one is the minimize the oxidative stress in case of the goat, and lowering not only minimize the oxidative stress, it also lowering the malondialdehyde that and boosting the antioxidant enzyme that I discussed in the previous slide. That is superoxide dismutase catalase and glutathione peroxidase. This is the powder form, and powder form that the brown color of powder. This one may be supplemented in the diet of the goat. This is the hematococcus from which astaxanthin is obtained, and this one is the Pythia yeast that is the also used commercially used in the formation of the astaxanthin that contains about 2%. This is the supplemented antioxidant, that is the beta carotene, vitamin E, astaxanthin, and vitamin C. How these vitamins hunt the free radicals. This one is the beta carotene, always found in the, this one is the actually lipid bilayers. And this beta carotene hunts only those free radicals that are formed in the cell membranes only. Because this one is the lipid soluble, beta carotene is the lipid soluble, only hunts only that free radicals that are generated in the cell membranes. And vitamin C is acts as an antioxidant. This one is the water soluble, and only disposes the free radicals that are found in the cytoplasm extracellular. This one is the vitamin E. This one is the vitamin E. This one is the decompose or neutralize the radicals that are found in both the conditions. But astaxanthin is the very important. This one is the most potent, and both sides. This one is the outside, both sides the plasma membrane, and it acts in plasma membrane itself also, and cytoplasm and extracellular radicals are also neutralized by the astaxanthin. This one is the very important in the mechanism of the action. Polyphenols. This is the most recognized polyphenol in the classes of flavonoids. This one is the silamorin. Silamorin is the obtained from milk thistle plants, and curcumin, that is the obtained from the rhizome of turmeric. This one is the may be supplemented in the diet of the goats, because these are the curcumin and silamorin, both are acts as antioxidant and neutralize the free radicals, and also promote the formation of the endogenous antioxidant enzymes. And this one is the potent activity to neutralize the superoxide anion and hydrogen peroxide, lipid peroxide, hydroxyl radical, and several reactive nitrogen species are neutralized by the silamorin and rhizome of the turmeric. Curcumin can also increase the curcumin is the obtained from the turmeric, can also increase the glutathione in the cellular level. This is the pictures of the milk thistle, and this the fruit and seed of milk thistle plant contains about the 60% silamorin, and turmeric rhizomes contains about the 1.5 to 3% the curcumin, and both are the both may be supplemented in fruit form. These are the antioxidant minerals. These are the antioxidant minerals that are the responsible for the activity of the antioxidant enzymes. These are the dose level, maybe dose level may be given to the in the diet of the goats, and this one is the actually zinc is the acts as a prosthetic group or promote their action necessary for their action. Without the zinc, the activity of the superoxide dismutase is impaired. And this one is the copper. Copper and manganese both are the also essential for the activity of the superoxide dismutase, and their level maybe these are the levels may be added in the diet of the goats to prevent the formation of free radicals and increase the productive and reproductive performance of the goat. And selenium, this one is the selenium. Selenium is acts as the selenium acts as a cofactor and cofactor for the glutathione peroxidase and also necessary for the action of the glutathione peroxidase. And this one is the iron. This is the this dosage may be added in the diet of the goat and stimulate the function of the catalyst and also acts as the factor of the catalyst. This is the whole oxidative stress, how the dietary supplementation of antioxidants prevent the increase the productive and reproductive performance by decreasing the disease condition, either metabolic diseases or infective diseases. And this antioxidant maintains the homeostasis in the cells and tissue, and that cause better health and metabolic activity. And if the health is better and metabolic activity is better, then the performance of the reproductive and productive and growth performance will be better in the animals. And meat, if the meat that are obtained from the dietary supplemented of antioxidants, this cause delay lipid and protein oxidation and improve the quality and preservation, and by which these all are responsible for the extended the shelf life of the meat. And not only meat, this one is the also the goat that fed antioxidant. The milk of that goat is the reduce the production of the free radicals and also reduce the other infections and other infection. And these all the factors are responsible for the quality of the milk in the terms of the protein, fat, and somatic count, and increase the nutritive value of the milk because the protein damages by the free radicals is the low in antioxidant, and somatic cell count is the increase in the milk that have a low antioxidant level. This one is the very important for the animal, not only animals but meat and milk also. This is the whole. This is the summary how the free radicals generation takes place by the in the mitochondrium, peroxisome, pyrosome conditions. In these conditions, and how these free radicals are changed into the neutralized by the catalase and glutathione peroxidase and superoxide dismutase. And this the free radicals that start the chain reactions may be the neutralized by the chain breaking antioxidant. This one is the vitamin E and vitamin C are the chain breaking antioxidant that breaks the chain reaction that is takes place in the polyunsaturated fatty acids. This is the all. Thank you very much, sir.